This presentation on diaQTL is sponsored by the Tools for Genomics-Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 15, 2021.
Right, so we're going to start talking about linkage mapping in multiple F1 populations, and our focus here is on tetraploids. The name of the software is diaQTL, which if you don't get it immediately, it's a contraction of the words diall and QTL. And my co-authors in this effort, we introduced them yesterday. Chaoji Zhang from Wageningen University, Patricio Munoz, and Rodrigo Amadou from the University of Florida. And for more information, you can check out the bioRxiv preprint. So yesterday we talked about PolyOrigin, which is the software that will analyze the genetic marker data for your diall and then output genotype probabilities, and then those can be used in conjunction with the phenotype data to do the QTL mapping, and that's the purpose of the diaQTL package. And just a reminder, when we say diall, what we're actually envisioning is partial diall designs, which can range anything from a half diall to circular mating to factorial designs where you've got different sets of female and male parents. So it's very flexible, and that allows us to use this in conjunction with breeding programs to, I think, leverage the kinds of data sets that we routinely generate as breeders. Now we can use them for gene discovery as well. That's the bigger picture here. So the package is maintained on GitHub. It is fundamentally working with a multi-allelic QTL model. So it's general by design, and that can be both a strength and a weakness, as we'll talk about it in a minute. The basic workflow is similar to what you've seen already. So we go through a process of trying to discover the QTL and then follow up to estimate what are the effects of the different haplotypes. It has the capacity to fit an additive model as well as digenic dominance and higher order dominance for tetraploids. So that's a bit of a unique feature compared to the other software available. And it also will include a polygenic effect, so you can estimate the contribution of all these different types of genetic effects to your total broad sense heritability. It is set up to work with repeated measurements on the same clone over different years or locations so that you can do stage-wise analyses of multi-environment trials. And then besides the basic workflow to analyze the QTL, it has some auxiliary functions to help with visualizing and selecting on the haplotypes, and we'll illustrate that here in a minute. So the outline of the talk is we're going to look at some of the results from simulations and then go through the tutorial that's on the GitHub page. So if you're gearing up for that, you can follow the link to the GitHub site and prepare to be able to follow along in the vignette. So the simplest type of diall design beyond just a single F1 population would be these 3x3 half diall, so 3 F1 populations derived from a set of 3 parents. And conceptually, you can look at the problem sort of like this matrix. For every, in this case, I'm showing on the rows is the phenotype for different individuals, and each individual then has different haplotypes present. And the cells of this matrix then are the expected dosage of those haplotypes. And this, for an additive QTL model, those are the predictor variables. And every individual is going to have a sum of 4 because they're tetraploids. So if you add up across each row, you get 4. And sometimes you might have an individual which actually has a dose of 2 due to double reduction. And then the regression coefficients, so those dosages are the predictors, and then the coefficients are the additive effects that you estimate. And the output from the software, which comes from this function called fitQTL, it estimates the haplotype effects, and it shows it here with, so for each parent, in this case A, B, and C, there are 4 different haplotypes, 1, 2, 3, 4, and the sum of the haplotype effects is constrained to be 0, and so you're looking at deviations from that intercept for each of those haplotypes. And the error bars in this case are a credible interval, and you can specify what confidence you want to use for that. So this is similar to the type of model and results that you saw already from Guy's presentation. One thing to keep in mind is that each haplotype has its own the potential to have its own effect, different from all others. And in reality, we don't expect that, in fact, all of the haplotypes have different alleles at our QTL. In general, we would expect that some individuals have multiple doses of the same QTL allele. And this software at present doesn't have any capacity to automatically determine what is the true number of QTL alleles. But informally, at least, one can look at this and make a hypothesis that haplotypes which have a similar effect may contain the same QTL allele. So for example, in this is the output from looking at a particular trait in a potato data set, and maybe we can look and see that haplotypes 1 and 4 from parent A have an additive effect of around -1, which is similar to haplotype 4 of parent B and haplotypes 1 and 2 of parent C. So maybe those are all actually the same underlying QTL allele. Likewise, for the haplotypes that have effect around 1, there's this haplotype 3 of parent B, looks like it's more around 0. And this one here from parent C, haplotype 4, is about 0.5. Maybe those are different. At this point, with this kind of a data set, all you can really do is make these kinds of hypotheses. You need to follow up with sequencing in those regions to really know at the sequence level, are you dealing with how many different alleles are actually present at this QTL. And that is actually one of the goals of our SCRI project, is to look at combining the output from these types of QTL models with sequencing data in pedigreed populations to see if we can't provide the support at the sequence level to really identify which haplotypes are distinct QTL alleles. So that's the output for an additive model. What about dominance? So in polyploids, there's more than one kind of dominance. The maximum order of the dominance is given by the ploidy of the species. So in a tetraploid, you have digenic, trigenic, and quadrigenic components of dominance. And this particular decomposition in this way, and the one that's using the software, is probably not the kind of dominance that you're used to thinking about. Complete dominance, as we'll see in a minute, is a slightly different beast, but this type of a decomposition has nice statistical properties. In particular, it's what's called an orthogonal decomposition, and it's the classical method that has been used in quantitative genetics, for almost 75, 80 years now. This table attempts to give you some idea of what do these higher order types of dominance mean. So the first column, this is the name, additive, digenic, etc. And as you can tell from the prefix of the word, basically it's telling you how many copies of the gene are involved in that particular effect. So for an additive effect, the predictors are haplotypes. For a digenic effect, the predictors are diplotypes, etc. And here, this column, third column, is just showing you specific examples. If the parents are A, B, and C, the nomenclature we're using a period and then a number after that to indicate whether it's the first, second, third, or fourth haplotype of a parent. So these are all examples of haplotypes. If you add 2 haplotypes together, you get a diplotype. Put 3 together, you get a triplotype, etc. Finally, just to give you an idea of the combinatorial explosion that happens, how many actual parameters or effects have to be estimated for this even somewhat simple case with only 3 parents, you have 3 parents times 4 haplotypes, so it's 12 different parameters for the additive model. You have 78 for the digenic model, 240 for the trigenic, and 300 for the quadrigenic. So you can see the complexity of the model starts to increase quite a lot as you start to add dominance to the model. The other point here is that when we talk about a particular model being referred to as, say, digenic or trigenic, as opposed to talking about the effect, when we talk about the model, it's implicit that all of the lower order effects are included. So the digenic model includes the additive and the digenic effects, and the trigenic model has additive, digenic, and trigenic effects. So you always have the complete set of effects going up to whatever order you name the model. Now, I mentioned that this notion of dominance is a little bit different than probably how you're used to thinking about it. Say if we take, for example, the idea of complete dominance, which you can generalize to a tetraploid pretty readily. So if we said A was dominant to B or completely dominant to B, that means that the 4 genotypes that have at least one copy of allele A, they all have the same genotypic value, and it's potentially different than the genotype which has 0 copies of A. This type of dominance is called simplex dominance in the GWAS poly package, which is what I'm going to be talking about next. And some of you may be familiar with that already. The nice thing about modeling dominance that way is that you can describe it with a single parameter, which describes the difference between the 0 dose and the 1 or more doses of A. There's only one parameter there. But again, that's not the way it's being modeled in the diaQTL package. If you actually wanted to have a trait with complete dominance, you would have to have the most complex type of dominance model all the way up to the quadrigenic component. So there are some trade-offs to modeling dominance this way, but from what we've seen so far, and you'll see an example of a trait with dominance in a minute, the way it's being partitioned in the diaQTL package is working just fine. So how do we visualize dominance effects? We don't attempt to visualize the trigenic or higher, but the digenic dominance, which is a property of a combination of 2 haplotypes or a diplotype. You can visualize it using this kind of interaction plot. So each row is a haplotype and each column is a haplotype, and then the cells are the combination of those 2. So this is the visualization you get from this fit QTL function. Above the diagonal, those are the digenic effects themselves, and the color bar here shows what the numbers correspond to those colors, excuse me. And below the diagonal is the sum of the additive and the digenic effects. So basically it means that you add the same value for every row, and then that's the additive portion. And then you have differences corresponding to the digenic effects for cells between cells in the same row or column. And the visual effect of this is that you see how much more intense the reds and the blues are below the diagonal when you add the additive and the digenic effects together. And this is the visual cue that tells you that the additive effect is accounting for a larger proportion of the total variance. That's why you see these larger deviations from 0. And the software actually gives you the proportions of variance numerically. And in this case, the additive variance is 1/4 of the total variance, whereas the digenic dominance variance is only 9%. So we're going to see that again when we get through the vignette, but this is just a preview of that idea. Let's now start talking about the basic elements of the workflow, the first one being the QTL discovery. Before we can fit the QTL, we have to know where they are. And the name of the function in the package that does this is called scan1. You'll see a number of the functions in this package are pretty much blatantly taken from the names of functions in the original RQTL package developed by Carl Broman, just because it is such a great piece of software that many people are familiar with. So we've used that to model some of the functionality of this package on. So it's going to calculate a LOD score at each marker and present them in this type of profile that you can see. And you heard about LOD scores already from Chris in the first talk today, so it's exactly the same idea. And the real question boils down to what is the threshold that you're going to use? And the threshold needs to be chosen so that you can control the probability of making a false positive or the false positive rate. And there are two main ways that that you can do that within this package. The permutation test you heard about already from Chris. It's the main downside being that it's computationally intensive. To do it, you need to do it usually about 1,000 times. And the alternative is to do some simulations without any QTL. And that's actually that's the approach that we've taken. And the nice thing is that because any population you create that's a diall is more or less going to conform to certain types of genetic properties, you can do the simulations generically, and it should hold for it should hold for everyone else's experiments. And you'll see when we talk about GWAS, that's not the case just because there's no real regularity to the kinds of populations that people use in GWAS. But for linkage mapping, the simulation approach is quite convenient. So here is one of the figures from our paper, which shows what was the impact of different types of parameters on the design of the diall, including the ploidy. So we're going to compare a diploid versus tetraploid. This software works for both ploidy levels. The number of parents was varied from 2 to 8. And then we also varied the size of the genome. And for each one of those different scenarios, a trait was simulated without any QTL, so just noise basically. And then the question is, okay, well, what's the maximum LOD score that you observe when there is no QTL in the data set? And taking the 95th percentile of that gives you what threshold you should use for controlling the significance level at 0.05. So you can see that the threshold that you need to use increases as your genome size increases because you're doing more tests. And so you have more possibilities of making a false positive. As you increase the number of parents in the diall, so from red to purple goes up, we also have more higher LOD scores are being observed just by chance. And so you have to increase the threshold for that reason. If you look at, say, the gap if you go from 2 parents to 4 or 4 to 6, this solid lines here, this is the diploid case, but similar when you look at the tetraploid case, it's almost linear. It's actually not quite, but the change in the LOD threshold as you go from 2 to 4 is almost as much as, it's slightly more than the difference as you go from 4 to 6 and so on. And again, if you look at the dashed lines at the top, you can see that the LOD threshold for a tetraploid diall is quite a bit higher than the LOD threshold for a diploid diall. And so for this example that we're talking about, if we had a genome of 10 Morgans and we have 3 parents, that's somewhere in here, we're looking at somewhere around, say, a 6.5 LOD threshold if we wanted to keep our alpha at 0.05. Now, these simulation results are accessible to you in the package through a function called LOD thresh. So you just give LOD thresh the size of your genome, how many parents, and your ploidy, and it tells you what LOD threshold you should be using. The other main important feature that you want to characterize when you talk about designing an experiment is statistical power, right? What's the probability of detecting the QTL when it exists? And this is the result of the simulation. Again, we've got a bunch of points here, and then we've got 2 different splines to the data. So the solid line is the spline for a QTL with a heritability of 0.2, and the dashed line is the spline for a heritability of 0.1. And this is over a couple different numbers of parents and varying the population size. And what we found is that across these different types of designs, you can largely explain the power based on this metric, which is the progeny per parental haplotype. So if you take the total population size divided by the number of haplotypes in the data set, which is your number of parents times your ploidy, that more or less is a good predictor of the power for a given heritability. If you look at this closely, you'll see that the diploid and the tetraploid scenarios don't quite line up on the same spline. There is definitely a slight difference there, but it's close. And of course, as you go from a low heritability of 0.1 up to a heritability of 0.2, you get higher power. So if we say we wanted to have a power of 0.9, we want a 90% probability of detecting the QTL. If your heritability is 0.2, then you only need 20 progeny per parental haplotype. And that number increases to about 50 progeny per parental haplotype if you're talking about a heritability of 0.1. So that can help guide your thinking about, okay, how do I design some QTL mapping experiments for these types of multi-parent populations? Besides detecting the QTL, we'd like to be able to estimate the effects of the haplotypes accurately. And so this figure shows what is the correlation between the simulated and estimated effects. Again, using the same kinds of regression methods, we can fit a spline for the high heritability or the low heritability. And these two arrows here just show you what were the PPH or progeny per parental haplotype values we were talking about a moment ago. If you have a 90% power at a heritability of 0.2, you have to have 20 PPH, and that would correspond to an accuracy of 0.9. And it's about the same for the low heritability. So by the time you get to a power of 0.9, your accuracy of your haplotype estimates is around 0.9, which is quite good. All right, so let's talk a bit about how does it work. Due to the large number of model parameters, we saw even for 3 parents, as soon as you get to even a digenic dominance, you're already at 78 different effects to estimate. That we really need to use a random effects model, and Guy already gave a good motivation for the value of doing so. So we're kind of following in the footsteps that he helped to develop that concept. The difference is that instead of using a normal distribution, we're going to opt for some non-normal distributions. And to do so, it's typical to rely on a Bayesian regression methodology. And rather than reinvent the wheel, we're going to use some software that was already developed to handle these types of non-normal distributions for genomic selection models, where you fit all of the markers in the model at the same time, and you want to allow for the fact that some markers may contribute more variance than others. And so in this case, we're only fitting one marker, but the idea is that we want some haplotypes to have a much larger effect than others. So these non-normal distributions are a little bit of a midway point between a fixed effects model and the typical normally distributed assumption for random effects models. The name of that package is BGLR. And because the BGLR is using a Monte Carlo method that is used for these types of Bayesian models, we have to come up with an appropriate number of iterations for the algorithm. And I'm far from an expert on Monte Carlo methods for Bayesian regression, but we've tried to create a function that can help you in a simple way decide what is an appropriate number of iterations, and that function is called setparams, which calls some other packages that are designed to look at properties related to convergence and autocorrelation, some of these other things. So setparams, you give some information about what you're trying to do, and it will then tell you how many iterations you need to use. And we'll see that here in the tutorial. Lastly, as we start to look at different genetic models at a particular QTL, we're going to be relying on a measure of fit that's called DIC, which stands for the Deviance Information Criterion. It's like AIC or BIC in the sense that a lower value is better. And this is the type of penalized likelihood that is provided by BGLR. And it allows us to look at this trade-off between the model complexity and the model fit. So then let's talk a bit about, see how the software works. So it's on GitHub. Here's the landing page. You can see that Rodrigo is the co-author with me on developing this software. And you've got links here to PolyOrigin, the vignette that we're about to go through, and the reference manual, which provides more information about the syntax for these functions. One thing to just point out that this is still a package that's under development. The basic functions that we're going to talk about today are more or less finished, but some of the auxiliary functions are improving. And in fact, just a couple days ago, Rodrigo modified the way we run the multi-core algorithm, so it works on Windows now. So the current version is 0.91, and if you are interested in checking back regularly, you might find that there are some new functions to look at. So the Vignette dataset is a 3x3 half-dial L from Potato. It's the same data set that was used in the demonstration for the poly origin. And the trait we're going to look at is length to width ratio for potato. So these potatoes are all from the red market category in the United States and in Canada, in which case you want a fairly round potato. The commercial varieties typically have length to width ratios around 1.1, 1.2 at best. And you can see here that in these largely unselected populations, we have length to width ratios as high as 2. The input files, there are 3 of them that are needed to run the software. The first 2 are the pedigree and the genotype file, and they basically come directly from poly origin. You heard from Chowji yesterday about there's a file that's produced by poly origin that has the word poly ancestry in it. And this function within the diaQTL package called read poly ancestry. It takes one argument. You just give it the name of the file that you got from poly origin, and it will parse that file and give you the pedigree and genotype files that you need to run diaQTL. The phenotype file you have to prepare separately, and we'll look at that in a second. So the pedigree file structure is quite simple. It's 3 columns, your clone name, and then the 2 parents. The importance of using this read poly ancestry command is that the order of the parents is not necessarily mother and then father. It has to correspond to the order of the parents that is used internally when poly origin does the analysis. Otherwise, the coding of your haplotypes is not right. So just use the read poly ancestry and you won't run into any problems. This one line of code right here just is a quick way to see, okay, how many individuals do we have in each of our populations? So we've got, over 100 up to 155 for the 3 different F1 populations. And this command right here, the system file, is just a quick way to ask R to tell you what is the name of the file with this name from this package in this subfolder, and it generates the complete path. So that way, no matter where you've installed the package on your computer, you'll get the right file name. The genotype file, it has the first 3 or 4 columns, depending upon whether you include a physical reference genome. Those correspond to the map. So the rows are the markers, and then after the map, the subsequent columns are all the genotype probabilities that you're going to get out of poly origin. And poly origin by default will produce the genetic map in the output, and you have to have that, the centimorgan map. But at least personally, when I like to look at a lot of my QTL results situated against the reference genome to better understand what some of the candidate genes may be. And so you can also merge back into your map the reference genome that you may have used originally to order your markers for poly origin. And you'll see that's one of the options when you run the read poly ancestry command. So that's our genotype file. Lastly, the phenotype file. The simplest possible phenotype file just has 2 columns, which is a clone name and then a trait. Now you can have multiple columns for multiple traits, and then after your traits are listed, you can have columns for all of the covariates that you might want to use. For example, year or location, or even if you have a numeric covariate that you want to use, like you want to control for maturity or something when you analyze yield, for example, you can use those kinds of numerical covariates as well. Just to show you what our data look like, because the residuals, if we use the length to width ratio as is, we get a non-normal distribution for our residuals. We've used this log transformation to overcome that. One, I think, tip I like to pass on, and we heard a little bit about this from Guy's talk, is that students often misunderstand that they think their phenotype should be normally distributed, and they do a check of a histogram of their phenotypes. And in fact, phenotypes should not be normally distributed. They should only be normally distributed conditional on the QTL. The ideal case is that you'd have a bimodal distribution. That's how you know you have a strong QTL. So when you're doing your check of your model assumptions, it's the residuals that should be normal, not the phenotypes themselves. Okay, so now we can come to reading in the data. So that was the first couple slides were just showing you what those files look like. Now this is the first function that's actually inside the package. I noticed when Guy was talking, it looks like it's the exact same function, read_data. There are only so many clever ways to come up with a function with that name. And you give it the name of the genophile, the pedigree file, the phenotype file. You tell it what ploidy you're working with. As I mentioned, this works for tetraploids and diploids, but that's it. There's no plan to extend this to hexaploids. We're going to rely on North Carolina State's group to do that as part of our SCRI project. And then the other parameter here that's of note is this one called dominance. And we've talked a bit about dominance already. There's up to this so-called quadrogenic dominance when you talk about a tetraploid. And the way we represent that is we always call the parameter dominance, and the value of that argument can be 1, 2, 3, or 4. So if you say dominance equals 1, it may seem a little bit odd at first that actually that means it's an additive model. You might think, well, shouldn't you say dominance equals 0? Yeah, from a certain perspective, that's true. We could have called this argument order or genic or something like that, but we just chose to call it dominance because it's the way you change the level of dominance. So the number that you use here corresponds to the number of haplotypes in the predictor. So if you have 1, that means you have a single haplotype, it's additive. 2, you have 2 haplotypes, it's a diagenic. So hopefully that is not too confusing. And then lastly, you can specify how many cores you want to use to take advantage of that. The other note that I have here is to just clarify that what you specify here when you read the data does not imply that that's the type of model you're going to use in all your subsequent analyses. When you actually do the scan or you fit the QTL, you have to additionally say what dominance do you want to use in that analysis. The point of the dominance here is that it's going to set the upper limit for what kind of dominance you can use in the subsequent analyses. The reason for that is that the data, the input file, the genotype probabilities is condensed in a certain way that you can't undo when you read the data. And that's for storage reasons. And whatever you decide to do, that's going to limit the maximum possible dominance that you can use later on. So if you want the most flexibility, you would use dominance equals 4 when you read the data, and then you have the option of comparing additive up to quadrogenic dominance when you do your analysis. The reason you wouldn't do that is because of the additional time and space it might take if you have a big data set. And because it's probably pretty rare that you're going to have traits that are going to require a trigenic or quadrogenic model. We've just by default set the dominance at 2. And of course, you can change that if you want to. So when you run this command, it takes a little while because again, it's processing the probabilities into a more compact format, but it will tell you then, okay, how many individuals are in common between your pedigree, your genotype, and your phenotype input files. In this case, there's 413. The next step is to set the parameters for your regression models with using the Bayesian methodology, and that function is called setparams. As I mentioned, it's relying on some other packages that do this quite well. The one we're using here is called CODA. And the CODA package has this thing called a Raftery and Lewis diagnostic, which we don't need to understand too deeply. But the point is that you have to give it these 2 different arguments, and they're called Q and R, and those symbols have nothing to do with allele frequency or recombination. It's just the symbols that are what that particular package is using. And the Q represents what quantile of the distribution you want to estimate, and the R represents sort of what the interval around that desired quantile you want to estimate with a 95% probability. The practical advice that we have for you here is when you run your scan 1, just choose Q equals 0.5 and R equals 0.1, and that seems to give good results when you want to do your genome scan. So that's what we're showing here. If you pick those values, it's going to recommend that you use a burn-in of at least 20 iterations and at least 500 and so iterations. These are these values are generated from a Monte Carlo process. So each time you run this, you'll get a slightly different answer. But the practical advice again is pick something like around 50 burn-in and 500 total, and you should be fine. The other kind of preliminary is to see what that detection threshold should be to do your scan and to interpret the scan. And as I mentioned already, we've done these simulations and you can access them by using this lod thresh command. If you want to know, well, how, in one line, if you want to know, well, how big is my genome anyways, you can just use the tapply command and calculate the maximum, what's the largest centimorgan value for each of your chromosomes, add them up. And in this case, for the potato example, you get 12.1 morgan. So recall lod thresh, we tell it our genome size is 12.1, we have 3 parents, and we're a tetraploid, and it says, okay, use a threshold of lod of 6.8. If you want to run the permutation test, that function is called scan1 permute. So now we can do our single QTL scan. It's called scan1. You give it the variable data, which is what we read in from after we read all those files, what it was created, the name of the trait, the parameters that you estimated using the set params for the iterations. And then if you have multiple cores, you can specify that here. And what you're going to get as output is basically a data frame or a table that has for each marker what the result, what the lod score and some other information was from that test. And rather than showing you the entire thing, let's just look at what happens when you run this summary command. So there's a command called scan1 summary. You give it the full data frame that you got from scan1, and it's just going to look and say, okay, for each chromosome, what was the most significant marker on each chromosome? And again, this basically mirrors the kind of output you get from the original rQTL package, where it would just tell you what the peaks are on each chromosome. So we call that peaks. The scan1 summary function returns a list, and you access the peaks element using that $sign. So in addition to the lod score for each marker, you get a little r squared. It's not a big r squared. And the little r squared, because it's a squared correlation between your predicted and observed response. As we already heard a bit about, when you start to get to random effects models, then the notion of what the people are used to seeing for a capital R squared is a little bit different. And this is one way of trying to present something similar for a mixed effects model. The other column is called delta DIC. Now, I mentioned what DIC was earlier. It's the change in the DIC relative to the no QTL model. So a more negative number is better. If you look at this, you'll see that the chromosome with the biggest effect is 10. It has a lod of 39, an R squared of 37%, and the delta DIC was minus 161. You can also use the function. Besides giving you that table, it also generates a nice plot that you can use for a publication. And it shows you the lod versus all the markers, and it uses a solid line to connect the results. So those are the two main outputs you get from the scan1 summary function. Now, you might want to go beyond just knowing what is the marker that had the highest value. You might want to know what the potential interval is, and there's a function called Bayes CI, which gives you a Bayesian credible interval, and it uses the same idea present in the QTL package. It basically takes the lod score and looks at what area under that lod profile for that chromosome accounts for whatever percentage of the total area. And that CI prob this would mean that you want to know, okay, 90% of the area under the lod profile is between these two markers. And yeah, so in this case, the lod profile goes from about 48.2 megabases to 50.8 megabases. So we have something like a 2.5 MB interval, and this happens to coincide with the location of a well-known QTL for potato that controls length to width ratio. And now we even know what the what the actual gene is. So that's the value of the Bayes CI function. Now let's transition from the QTL discovery to actually fitting a QTL model at the peak to try and understand the haplotype effects. And we want to now run the setParams function another time. The reason being that the first time we ran setParams, we didn't tell it, we didn't give it a particular marker. We were trying to find a value that could work across the whole genome. And we also didn't necessarily need to estimate the tail of the distribution very well from the Bayesian posterior distribution. But now that we're actually trying to drill down on a single QTL, you're going to want to probably use more iterations, and you can afford to do so because you're only doing it, once instead of thousands of times. So if you want to use, say, a 90% credible interval for your haplotype effects, one rationale would be to say use a Q of 0.05 and an R of 0.025. Again, those are just kind of recommendations. You can play around with that if you want, but now we can also tell the setParams to only focus on this marker instead of trying to get a value that works across the whole genome. And we can also tell it, well, we want to run a dominance model, so do we have to modify the number of iterations for a dominance model? So you can tell that with the dominance equals 2. And you can see that, yes, indeed, with these new values, we have to use a lot more iterations. It says we need 26,000 iterations instead of just 600 when we did the scan. But it's still, it sounds like a big number, but it runs in under a minute on a normal laptop. The two different, here you see the two different calls to fit QTL. The first one is using an additive model. And the second one, which I call fit2, it uses this dominance equals 2 argument. So now we want to include the diagenic dominance, and let's see if it makes much of a difference. And if we want to compare which of these two models was best, we should look at the piece of information that's returned by the function is called delta DIC. So if you take the variable fit1 $delta DIC, and then the same thing for fit2, and you compare them, we see that the model with the diagenic dominance has a 13-point lower DIC compared to the additive model. So we would select the dominance model as the better model in this example. The fit QTL function will also return to you the proportion of variance for the different effects, and that's under this element called var. So we have the additive, the diagenic, and it will also fit a polygenic effect, and it uses a kinship matrix identical to basically what Guy described. We are leaving out the markers on the chromosome that the QTL was on, and it's using just the markers on the other chromosomes to fit that polygenic effect. But this can help you understand then what proportion of the variance is due to this QTL versus due to smaller effect QTL that you might need to use a genomic prediction type of model for. And other things that are returned by the fit QTL function, it will return some plots. So under the plots element of the list, you have an additive and a dominance version, and these are the types of visuals that we saw at the beginning. This is the haplotype effects for that QTL, and then these are the diagenic dominance effects and the sum of them. Looking at this, we would say that the haplotype which has the largest effect in magnitude is haplotype number 2 from this second parent. It's negative, so that means it's reducing the length to width ratio, and it's reducing it by the most. So if the goal was to select, the goal is to breed potatoes, red potatoes that are round, this is the haplotype that we want to have in our parents. And so that kind of segues to the next part of the vignette, which is, okay, well, how do I actually find progeny or select progeny that contain this haplotype? How would I do that in the package? And you can do that using a function called haploget. Not a very great name, but anyways, that's the idea. Get it returns a matrix of the dosages of the haplotype. So this haplos variable is a matrix, and the matrix corresponds to this particular marker. And this is a kind of visualization of what it looks like. It looks kind of like what I showed in the beginning from the regression model. Each row is a different clone. Each column is the haplotype, and then the cells of the matrix are the expected dosages, which are not only going to be integral numbers. I've shown them like they're ones and twos, but in reality, they can be fractional values because you're dealing with the output from a probabilistic model. And you can see that this is this is more of a idealization. The actual data, you can look at it if you say type a histogram command. Let's look at the column that has this name. So the names of the columns are the parent concatenated with 1, 2, 3, or 4, and a period separating them. So remember, this was the name of the haplotype that we just saw, is the one that makes the round potatoes. So let's look at the dosage at this haplotype across the population, and that's what the histogram you see in the upper right corner is. Most of the individuals, the majority, let's say, have 0 dose, they don't contain it. There are some that have 1 dose, and you'll see there's a slight bar here, which some value is a little bit over 1. So again, that's because these are probabilities. These are expectations from a random model, but you can actually see there's also a slight number of individuals which have a dosage around 2, which is possible because the poly-origin software can model quadrivalence and correctly predict double reduction. And if we ask the question, okay, which rows of the matrix have a dosage greater than, I'm picking 1.8, just to allow for a slight error in that dosage estimate. It looks like there are 3 individuals. It returns, this is the return. It says these are the names, and these numbers are just their rows in the matrix. So there appear to be 3 individuals which have 2 doses of this desirable haplotype. And if you want to, you should always typically try and visualize this, because sometimes you may get a little wonky results with your haplotypes, and you want to just do a visual confirmation that things look correct. And so the haploplot command in the package will generate this kind of a figure. You give it your data, you tell it which individual you want to look at, what chromosome you want to look at. Do you want the x-axis to be in centimorgans or base pairs? And then the marker is just for the benefit of putting a dashed line to help your eye figure out, okay, where's that marker, where's that QTL that I'm interested in? So this is the location of that QTL. And sure enough, we can see that there's this dark blue segment. This particular individual appears to have inherited both of the sister chromatid fragments for that particular haplotype. So that would be if all else looked good about that clone, that would be one that you'd want to use as a parent, because it would be increasing the frequency of that desirable haplotype in your population. So that's the end of the kind of basic workflow that you can do with the package right now. A couple footnotes here, one about multiple QTL mapping. And the functionality of the software for multiple QTL mapping is fairly rudimentary at this point, and we haven't really put it through the paces yet compared to some of the functionality that we just saw from Guy's presentation. But it does have the basic ability to fit a cofactor in the model when you're running the scan1 or when you're running the fit QTL. And the cofactor is just the name of another marker that you want to use, add in the regression analysis. When is that important? Theoretically, if you have multiple QTL on different chromosomes, because if they're additive, then the polygenic part of the model will take care of that. It's really when you have epistasis between QTL on different chromosomes that it becomes more important to use the cofactor for your discovery or fitting of the QTL model. And then when you have multiple QTL on the same chromosome, you can use cofactors, but I really like the scan2 functionality that's in the QTL package, and our goal is to create a scan2 function within diaQTL to make that possible. So look for that sometime this year. I've got a couple other ancillary ideas about what you can do with the package. So far, we've just been talking about quantitative traits, but we can also analyze binary traits with the package. Here's an example from an F1 population in potato where we were trying to map a late blight resistance in a clone that we knew was late blight resistant, but we didn't know what R gene it had. And so when you have a binary trait, you have to code it as a YN trait. That's just a reserved letter designation that the software knows to recognize as binary. So it will convert all the N's to zeros and all the Y's to ones. everything else follows the same way. You can do your scan one, and you get a log profile. You can fit the QTL model. And in this case, the potato that was late blight resistant was called Payette. And we see from the haplotype results that we know that haplotype 1 of Payette is the one that contains the R gene. It has this large positive effect, meaning that it's increasing the um, predictor of the GLM model towards a yes or resistance in this particular example. So the interpretation of the y-axis when you have a binary trait, it gets at how generalized linear models are handled. In this case, we're using a probit link function. And the last slide I have here is just to show you one other function that if you are working with a binary trait or something that you want to try and do some fine mapping on, you can use this function called fine map. And you give fine map the left and right markers of an interval that say you ran your QTL analysis and you know that the QTL appears to be between these two different markers. You can run fine map and it will find all of the haplotype switches in that interval. And then it will also show you on the right-hand side what the phenotype was. In this case, the N and Y show us, okay, which of these individuals are resistant, which are not. And you can use this to really drill down and start to develop some hypotheses about what your causal gene might be. All right, well, that's all I prepared, and I think we have some time for questions.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding and Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.